Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia, and I'm very proud to welcome you to this edition of our 2022-2023 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since 2016, the Foundations of Biomedical Data Science Seminar Series has focused on the use of data science methodologies, artificial intelligence, deep learning, and statistical modeling as they pertain to different themes in the biomedical and health sciences. This year's theme is Data Science and the Public Health Consequences of the COVID-19 Pandemic, where we will enjoy presentations from leading thinkers about the issues, promise, opportunities, and hurdles associated with understanding the secondary health effects of the COVID-19 pandemic. These include delayed health checkups, treatments, mental health, drug abuse, increased obesity, and the long lingering health effects associated with the SARS-CoV-19 virus. Participants selected in our Biomedical Data Science Innovation Lab program leverage these presentations as vital material for our culminating in-person grant project development workshop to be held at the Lodge at St. Edward's Park in Seattle, Washington in June of 2023. Today, I'm delighted to welcome our speaker, Sen Pei, from the Department of Environmental Health Sciences at Mailman School of Public Health at Columbia University. At Columbia, Dr. Pei is an assistant professor studying the effects of environmental, social, and ecological conditions on infectious diseases and develops mathematical models and computational tools, to address surveillance, forecasting, and control of both seasonal and emerging infectious diseases. Dr. Sen, fun fact, Dr. Sen likes traveling and exploring different cultures and food. And continuing with our 2022-2023 Biomedical Data Science Innovation Lab theme, today's lecture is entitled Impact of COVID-19 Pandemic on Influenza. The COVID-19 pandemic has disrupted the circulation of many respiratory diseases, including influenza. And in this lecture, Dr. Pei will talk about several studies which highlight the impact of COVID-19 activities in different countries. As always, we are streaming this lecture live and for recording via YouTube. If you're watching on YouTube, thank you for joining us. Our specially selected 2022-2023 Data Science Innovation Lab participants and alumni are encouraged to submit any questions via the chat feature in their Zoom sessions, where I'll synthesize these and ask them on your behalf in the last 10 minutes or so of the lecture. And with that, welcome, Sen. We're super excited to hear about your presentation.
Thank you so much for your kind introduction. Thank you for having me here. So today I'm going to talk about impact of COVID-19 pandemic on influenza. We will have a discussion of how COVID-19 pandemic has changed the transmission of influenza in the last 3 years, and also talk about what's the implication for the flu transmission in the next couple of years. So the talk will be structured within 4 different topics. The first one, I will broadly talk about pathogen interactions and some previous studies on this topic using data science and different statistical and mathematical methods. And in the second part, I will talk about empirical evidence showing how how influenza transmission was impacted by the COVID-19 pandemic in the last 3 years. And third, I will use an example to show how we can use mathematical models and counterfactual simulation to quantify the impact of COVID-19 NPIs on influenza in different regions in the US. And lastly, we will discuss and talk about what's the potential impact on future influenza outbreaks caused by the COVID-19 pandemic, right? Okay, let's first start talking about pathogen interactions. So we all got common cold every year. So on average, each person will experience 2 to 3 episodes of we call acute respiratory infections, which is also common cold, we know. But we know that behind every of those illnesses, there's a pathogen, right? So there are a number of pathogens can cause common cold. Those include many endemic viruses, which I have a list here. Those are common seen viruses can cause common cold, including influenza virus A, virus B, respiratory syncytial virus or RSV, seasonal coronaviruses. There are 4 different types: rhinoviruses, human metapneumoviruses, human parainfluenza, type 1 to 3, and adenovirus, and also others. So this is a part of the list of those endemic viruses that can cause common cold. But also there's novel viruses that are coming up to cause severe, we call it pandemic, right? So those are the most popular one could be the H1N1 in 1918, right? That was like more than 100 years ago. And most recently, there's 2009 H1N1 pandemic. But there's also other viruses from we call it even influenza, right? H5N1, H7N9, those are highly pathogenic avian influenza, which can cause severe diseases, but it's rare. And we also heard about emerging infectious diseases about SARS in 2003, MERS, and also SARS-CoV-2, which is causing the ongoing COVID-19 pandemic. So among those different pathogens, people have to think about whether there's interactions between those different viruses. One of the most well-known example is the interaction between influenza and bacterial infections. So here it's not virus-virus interaction, but it's a virus and a bacterial infection. Here in this figure, we can look at the number of incidences caused by influenza, shown by the blue line, and also the pneumococcal pneumonia, shown by the red line. So you can see both of them have very strong seasonality. They tends to peak in the winter times and have a lower number of incidents in summer times. But if you look at it closer, you will find it whenever there's a peak for flu, there's also a little bump for pneumonia incidents, right? So this is one of the example people have noticed a long time ago. They found that usually for those vulnerable people, they get infected by influenza. The mortality usually is not caused by the flu itself, but maybe the subsequent bacterial infection. And people have studied using mathematical models and data to study those interactions. And they found that flu infection actually can increase the susceptibility to subsequent bacterial infection. And this is one very classical example to show how we can measure pathogen interactions. And when it comes to virus-virus interactions, particularly respiratory disease interactions, here is a very interesting study from UK, and they look at the prevalence of pathogens among patients in several hospitals in Glasgow, UK, from 2005 to 2013. And you can see for some of the viruses, here is influenza virus A and the rhinovirus. They tend to have opposite phasing here. When the flu is high, rhinovirus goes low, so there's a correlation is negative. And also, if you can look at influenza B and adenovirus, there's also a pretty strong inverse pattern, right? When there's one is low, the other is high. But at the same time, there's some other pathogens like RSV and metapneumovirus. You can see there's a very strong similar seasonal patterns. Both of them peak at around similar time, and also PIV, parainfluenza virus 1 and 2. So those are all, those time series show that probably there may be some interactions happened across different viruses, right, between those different viruses. And then when we think about that, we always want to know what may be the possible pathways for pathogen interactions. And there's a very nice review paper by Apatovsky and colleagues, and they summarize the potential pathways at different levels for pathogen interaction. And those include cellular level interactions, host level interactions, population level interactions, right? So I can briefly go through all of them. Like at the cellular level, there may be competition for the resources for virus replication. If there's one virus infected one cells, the other virus replication could be impacted, right? There's also maybe cell damage caused by one virus, which can change the virus replication of others. At the host level interaction, it's mostly mediated by immunity or physiological changes. For instance, if people get HIV, it will weaken the immune system, which can lead to further infections, which otherwise would not be possible because of the immune effect, right? And also there's cross-reactive antibodies. if a person got infected by a very similar strain of virus, there could be cross-protection happening. So reduce the severity or maybe the chance of infection for that. There's also like interference signaling. For physiological changes, you can think about like fever, right? Fever is, people have fever, then you have a higher temperature, the replication of all the, maybe the viruses will be slowed down, not only the pathogen causing the fever. At the population level, this is more related to behavior changes, which can reduce the contacts and travels across different locations, right? It slow down the transmission at the population level. And also there may be medication use and vaccination that could change the likelihood of getting infected by other viruses. There are many different methods we can try to use data to identify pathogen interactions. I would argue that the most important way to do that is you can do some testing at the individual level. So probably you can have a multi-panel PCR test to see whether this person has this virus, right? And then you can use those very high resolution granular data coupled with some regression models, statistical models to quantify whether there's a significant interactions between different viruses. And on the right is a study from the UK I just mentioned a few slides ago, that they use the data, individual level data, which is really hard to collect and very expensive, I guess, to quantify the interactions. And they found there's evidence of strong negative and positive interactions between different pathogens. But this data is not always available, right? So people sometimes have to use surveillance data, which measures the time series of disease cases or other like maybe percentage positive to do those kind of analysis. And people have used multivariate Bayes and hierarchical models these are statistical model to disentangle the row. You can control a lot of variables to see whether there's interactions happening. But there's also other people using state space method reconstruction to try to use a framework of causal inference to see if there is there's a directional interaction between different pathogens. And also people can use other time series method detecting causality or correlation or even dynamical coupling between different time theories, right? And lastly, people can use some mechanistic model. You can assume there's an interaction term in your model and to fit the data, fit the model to the data to estimate how large is interaction there. So there are pretty bunch of ways we can measure interaction, but not all of them are working well given the data quality and all the limitations we have in those modeling methods. So yeah, so that's basically a brief overview of what people have done in terms of quantifying pathogen interactions. But here we want to look at what are the empirical data showing what happened for flu during the last 3 years. Okay. The first question is, how can COVID-19 impact flu transmission? I guess most of the important way we can think about it is that SARS-CoV-2 and influenza share very similar transmission routes. This figure shows the potential transmission pathways for SARS-CoV-2, and you can see many of them actually work for flu. And because we implemented very strong NPIs, either governmental or voluntary, right? Which has effectively reduced contact between different persons. So the chance of infection for both flu and SARS-CoV-2 has been reduced substantially. And also there's a potential effect of limited travel restrictions, right? You don't have that much travel from one location to the other, which can limit reintroduction of flu virus from one place to the other. There has been some studies to look at co-infections of flu and influenza, flu and SARS-CoV-2 at the individual level. There's one review paper. They gathered a large number of studies, and they found that only about 0.7% of people have co-infection confirmed by PCR test. So it's not very common to have co-infection. But also there are some studies using those data to show that influenza actually is associated with a reduced rate of SARS-CoV-2, right? But there's multiple different ways you can explain how this association happened, right? And okay, all right, let's look at the real world data in the US in the last in 2020. So let me explain a little bit what we are looking at here. So we are looking at the curve of percentage of visits for influenza-like illness. It quantifies what's percentage of people going to the hospital having symptoms like influenza. So it's not like a very specific metric for influenza because other viruses can cause similar symptoms. It's a symptom-based, but it can give you a broader sense of what happened over the year, what's general level of respiratory diseases. And we can see there are different curves from different seasons, and usually the IOI peak around the winter time, which we understand, right? And also here is a special case, which is 2009 H1N1 pandemic season. It peaks in the unusual time, very early in the season, in the autumn, in the fall time. And here the red line here shows the level of IOI in 2020, right? So this is so you can see during the COVID-19 pandemic, actually the activity of IOI is very low. It's suppressed by NPI and maybe other factors. So we are seeing very small number of respiratory infections during the first year of the pandemic. And there's also a study from Rachel Baker and other colleagues at Princeton to look at what is a positivity rate, right? You test a number of samples, you can look at what percentage of those samples have infected by influenza. This is a more specific target for influenza because you have to be confirmed by lab test. And you can see on the left, we show the percentage positivity in different states over time. And the vertical line here shows the timing for the NPIs in the US. You can see after that, you see a sharp drop of positivity rate. And those dotted lines are showing the previous seasons, the typical level of positivity rate. And here's a nice color plot, color map shows, you can see after the NPIs were implemented, there's a sharp reduce of positivity in all states of the US. And then when we look at globally, we can use data collected by the WHO FluNet to look at how flu activity changes during the COVID-19 pandemic. So on the left, we show the positivity rate. The positive cases, this is positive cases. On the right, we show the samples tested, both in Northern Hemisphere and Southern Hemisphere, because the seasonal is different, right? So you have to look at differently. And you can see before the COVID-19 pandemic, it's very clear seasonal pattern. In the winter, you have a lot of cases. But here, if you look at the number of cases after COVID-19 pandemic, it essentially goes to very low level, right? It's not, there's some data here that's very low. I'm showing a zoom in plot here. The number drops from, I think maybe the order of like tens of thousands to a few hundred. So like maybe only 1% of previous level. And people may ask, is this because of people are testing less during the COVID-19 pandemic? Actually, it's not, because here you can see the number of tests is still very high, right? It's testing a lot of people, but you don't see any of those positives. And there's some other studies looking at COVID-19, look at flu activities in different countries specifically. On the left, you can see this is work from China. Those blue line and the black line shows the positivity rate in the 2 seasons prior to the COVID-19 pandemic. But then after NPIs implemented in China, also the positivity rate drops significantly. It's much lower than previous season. And also you can look at data from South Korea, also has very strong NPI at the beginning, right? And those data, those dotted lines shows the previous seasons. And the black line shows the data in 2020. So there's a very strong suppression of flu activity in South Korea. Okay, now we have looked at some population level indicators of what happened for flu during the COVID-19 pandemic. Then we can think about how COVID-19 pandemic shaped the evolution of flu activity. Then we need to look at the sequence, look at the lineages and sublineages of flu viruses. There are four major circulating flu sublineages I'm showing here. This is from a beautiful realization from work from Hong Kong team. So they are taking the sequencing data from GIS-AID GenSet, which is the platform people upload your sequence data, right? We are looking at A, H3N2, A, H1N1. Those are two major circulating viral strains. And also B, Victoria, and B, Yamagata. Different colors means different strands, you know, depending on how you classify them, right? And the width of this band shows the number of samples within the data. So you can see clearly after the spring of 2020, I mean, you are losing a lot of diversity. Only most of them actually disappeared in the samples. You can only have a very small number of active circulating lineages observed in this big database. And surprisingly, has been no B Yamagata sublineage observed after spring of 2020. So you can see it's actually the NPI was so strong that it removed a lot of those circulating, strands, and only remains some of the major ones, probably in some locations with a higher transmission or maybe denser population can sustain their transmission there. And this can be better seen by looking at the phylogenetic trees for the four different sublineages, right? So you will see a lot of diversities of the strains before the pandemic. And after that, you will see a lot of them, they just disappear, only have a very small number, and only a couple of them survived entering into 2020 after. And you can see, strikingly, you can see the B Yamagata just disappeared. There's no sampling of that. So this has a pretty strong, this has also very pretty strong geographical patterns we can see here. This is also from the same paper from Hong Kong, which shows very, very informative how the evolution of influenza has been changed by the NPI, right? So for each of those sublineages, we have a comparison for pre-COVID-19 data and the COVID-19 data. This is from April 2020 to July 2021. Before that, you can see there's a lot of diversity, antigenic diversity, different strands circulating around different areas of the world, like for H3N2, right? But after that, you will see, you can see the majority of the strands, they just disappeared and only have two major strands circulating, one mostly in Southeast Asia and South Asia. The other one is in West Africa. So the transmission has been largely localized because the connection between different locations has been strongly reduced. There's no traveling across different locations. There's very limited traveling actually. So you will see there's a very isolated evolution of that. And this pattern also shows for other sublineages. Here is the figure comparison for H1N1, right? You can see used to be, across many countries around the world, but now you can only observe some major ones in West Africa. Those are the hotspots transmission. And for B, Victoria, also you can see the similar patterns. For the B Yamagata, you literally didn't see any samples after the COVID-19 pandemic. And there's a lot of discussions on whether there's a real elimination of B Yamagata strain. And this is because there are several reasons we can think about why that could happen. The first one is, after the COVID-19 pandemic with those strong NPIs, flu viruses can only survive in several hotspots. Some of the places there's a small population may not be enough to sustain those transmission. And also, the survival of specific strain is affected by the antigenic diversity. So according to WHO, as of February 2020, there were no confirmed B Yamagata detections since March 2020. So this is a huge, information, right? And the suppression of B Yamagata could be because of COVID-19 actually coincide with a period of low incidence and low antigenic diversity for B Yamagata. Even before the COVID-19 pandemic, the incidence of B Yamagata is very low, and also the antigenic diversity is very low. So they are very similar. And also probably it's because of people discussing about this. Probably it's because of prolonged use of a well-matched influenza vaccine, because in previous seasons, the matching for the B Yamagata, one of the component in flu vaccine is very well, because there's a lot of antigenic diversity. So there's still an active debate on whether there's a lack of detection or it's true extinction, which may have some explications on what we want to choose in the strains in our flu vaccine, right? And yesterday, actually, I took a look at the data from WHO FluNet. On the top, we show the data from the end of 2019 and until the most recent data. You can see they have different strains color coded here. This period, mostly in 2020 and early 2021, there's almost nothing reported, very small number of cases. But if you specifically look at B Yamagata, it's dropped significantly in the spring of 2020. And then there's some sporadic reporting there, but it's not clear whether those are actual transmission, or it could be sustained. Even in this season, it's a very small number, lower than 10 samples submitted across the whole world. So it's very interesting to look at whether B Yamagata will get back, or is actually eliminated. All right, then I think the other topic we want to cover is to use. We know that there's a lot of reduction of flu activity, but can we quantify the reduction? Can we get an exact number of how many infections were not happen because of COVID-19? But however, doing this actually is not an easy task. There are a couple of reasons, there are a couple of challenges for doing this. The first one is there's strong variation in peak timing and peak intensity across different seasons. As you can see from the earlier figure, I showed the curve for different seasons. Even though people know that flu typically peak in the winter time, but the peak timing can be vary, and the peak intensity is very different according to different factors. So you just cannot take the historical average and to look at the difference between the observed and historical average, because the historical average is not accurate for what happening in that season, specific season. And second one is the types and subtypes with having very different seasonal phasing. Not very different, but there's some difference. So we need to estimate A and B separately. And then we can think about there are many factors different. There are many differences in the geographical locations, right? For instance, there's some regional dynamics, differential MPIs implemented, and also maybe the initial susceptibility could be different at the beginning for flu, right? So we want to estimate those numbers for each region in the US. And here in this study, we use the HHS region level AOI plus, which is a metric we use. We multiply influenza-like illness by laboratory test positivity rate for influenza A, H1, and B. We are not considering H3 and 2 because there were basically no H3 and 2 in that season. So we will mostly focus on A, H1 and 1 and B. So how do we simulate transmission of influenza? And people have been using mathematical models. We know a lot about the SAR model during the last few years, right? So it's a similar idea that we can use those type of dynamical models to simulate the influenza-like illness plus signal we have here. And this is a model structure here. It's a SARS model susceptible, infected, recovered, and susceptible again model. But the transmission rate here, beta, is changing over time. We impose some seasonalities using humidity data, because we know humidity can change the transmission rate for influenza. In winter, when it's a low humidity, you have a higher transmission, so you can see more cases in winter. And this is a very simple model, right? We want to calibrate the model to the real world data using MCMC method to estimate parameter and initial conditions in different locations. Okay, so before we do anything to the using the real world data, we first want to see whether this model can actually capture the transmission, the disease data, right? So we did some retrospective forecasting and fitting using historical data here. So in those places, in those figures, we are feeding the data to AH1 and B from previous season. This is a national data, but we also have regional data. And the blue dots are the reported data, actual observation, and the gray area, the gray line is the mean prediction, mean fitting, and the gray area are the 95% confidence interval. So you can see mostly the model can reproduce the general seasonality and epidemic curve here. And then we apply the model to the real world data in 2019 and 2020 season. So you can see here, I'm using the vertical dotted line to show March 15th. That's the week when there's a strong, I think, lockdown happened in New York City and also in other locations. We fit the data until this time point. We only use the data before this time point to fit the model, and we can project the model into the future to see what if there were no NPIs, how many cases were reported, right? So it's basically a counterfactual simulation showing that, okay, we assume this model can reasonably represent flu transmission, and we want to see, without any interventions, what would happen in the spring of 2020. And then you can compare those projections with those actually observed data point here. So you can see there's some difference here, right? But because of the NPI happened at the tail of this season, so the absolute number of reduction actually is not that much, right? And this is particularly true for B, because it's already goes very low by the time of people implementing NPIs. Then we can quantify the reduction of influenza incidence compared to the predictive values within 10 weeks after March 15, 2020, right? It's a long sentence, but basically you take the predicted value, you minus the observed value, right? That's the absolute difference between the predicted and the observed. And then you can divide this number by the predicted value. So you will see the percentage drop during the 10 weeks period after lockdown. And we saw some variations in different regions in the US. Can see some of them, they can be reduction by maybe 70%, but some of them, they can be up to 90%. And for B, it's even larger. Some of them have a low of 30% reduction, probably already the activity very low. But some locations you can see there's reduction of up to 90%. So there's some geographical distribution happening here. And we didn't include region 2 here. Region 2 is including New York State, New Jersey, and also somehow Virgin Islands. So I don't know how they classify, it's very different, but they classify them somehow together into region 2. But we didn't include that because we found there's abnormal reporting of influenza like illness in region 2. So this is data from the flu view. The percentage of people going to see doctors with influenza-like illnesses. And you can see after the lockdown here, you actually see a lot of more reporting of influenza-like illness in region 2, mostly driven by New York area. I think that probably driven by the behavior change, right? People got panic, they want to go to hospital to see if they got COVID or flu or other respiratory diseases. And then you can see at the bottom, we show the number of positive specimens collected during this period. And you can see there are actually no increased number of specimens tested during this period. So I mean, that's why the reason, when we quantify those, you have to consider some issues within the data. There's abnormal reporting there. So we eventually did not include this region in our study. And lastly, the last slides I show, we quantify the number of reduction in terms of incidence. But what is reduction of transmission rate, beta in the term, right? So we can vary beta by some multipliers, and you can find the best fitting beta and compare that with, to see which is best fitting and to explain the observation of incidence after lockdown. And we found that the transmission rate has dropped significantly across different locations. And in the US, for H1, it's dropped by half. For B, dropped by maybe 1/3. So those are some methods we can use to quantify the impact of the reduction of the impact of COVID-19 on flu transmission. All right, so yeah, those are all the data we have in the past. We do some analysis retrospectively, but another big question is what will happen for influenza? What do those means? What do those means in the future for flu outbreaks? And people have done this also, right? You can use a model, for instance, here in our paper, we have projected the model into the future using the fitted model, giving some predefined scenarios, right? For instance, here we are assuming, okay, how about we maintain the same level of control for 1.5 years, and then you lift the control at this point, what you would say in the next few years, right? So we are trying different levels of immunity. Like this means the immunity for influenza can last for 2 years. This is for three years and 4 years. We can try different scenarios to see what is looked at, but generally you will see is after 1 or 2 years of no flu activity, usually once there's a remove of NPIs, you will see a huge spike after that in that season. And that will have some ripple effects on seasonality here, right? So you have to take some time to reach back to the typical seasonal pattern. There should be some, and this effect depends a lot on the length of immunity for influenza. And there's another study, I just mentioned from Princeton. They look at the accumulation of susceptible people, right, during this time. It goes much higher. So usually in the previous season, when there's a new infection, the susceptible people will bring down, and then during the summer it goes up. When it goes down, there's a seasonal pattern here. But in the last 1 or 2 years, when there's no infection, there's a buildup of susceptible people. So once there's a removal of those control measures, you will see a large spike here, which is far higher than previous seasons. But here they are modeling RSV, another respiratory virus, which is also very important pathogen. But the basic message is similar. You would expect some huge spikes if you suddenly remove the NPIs. And then let's look at what happened. Now it's like 2023. People have dropped all those, most of those NPIs in, I guess, maybe in the last 2 years. So you can look at what happened in the season of 2022 and 2023. That was last winter. We have seen a very large and early outbreak for influenza, is for IOI. And usually at this time, you don't see that high activity because you need really go into the winter to see the ramp up of influenza outbreaks. But in last season, we are seeing very early onset of flu activity, and also the peak is pretty high. As I said, this is IOI. It's not specific for influenza, but it has some indications of what happened. And then here, there was an article from New York Times, they're talking about the triple-demic happened in the winter of 2022. They look at COVID-19, RSV, and flu activity. And here they are using hospitalization data, so which is more specific. It's definitely for flu because it has to be confirmed for flu. You can see COVID-19 actually is lower than previous season because it's Omicron, and there's a very lower severity, so there's a smaller number of people going to the hospital. And RSV goes much higher. Those lines are the previous historical data. It surpassed, I think it's the highest level in the last 10 years, probably. And most of the hospitalization are actually for young children who have never experienced RSV before. And for influenza, it's much higher, right? It kind of like it surpassed the hospitalization caused by COVID-19. So we do see there's a large rebound. So the model kind of tell us the qualitative result that there will be some severe outbreaks happening after NPI were implemented. But what will happen this year or next year is still not very clear, I guess. Another big topic here is a disruption on virus evolution, right? So we can see there's a huge reduction in antigenic diversity, and which brings in a lot of uncertainty in how the flu virus will evolve in the next few years in the future. The first we can see, the uncertainty comes from the isolated geographical distribution, and those may lead to independent evolution, right? So there may be some, some location-specific strains appear. And then because of different prior infection history, there may be a diverse population immunological landscape, maybe in US, in China, in different countries, they will impose different evolutionary pressure on the evolution of viruses. And then you can think about there's a huge difference in vaccination for flu. So that's another level of uncertainty. You don't know what are the pressure of for evolution imposed by vaccination, right? And this has a very significant impact for the vaccine virus selection here, because if you found usually now we are using a very similar formula for the whole world, but if you found that okay, maybe this area has a very specific viruses strains for this location, how you can design a more effective formula for the vaccine. You have to consider those, right? If there's a co-circulation of antigenically distinct lineages, it imposes a challenge on how you select the formula, the virus in the vaccine. And the other thing, the other topic I will briefly touch on is because of there's a lack of flu outbreaks in 2020, 2021, it may have some implication for children because they don't have initial exposure to influenza at their early age. There's concept they call it immunological imprinting or original antigenic scene. This figure shows some of the ideas. I'm not expert on this, but I can generally talk about what the general idea here, right? So the x-axis shows the age of the person, and the y-axis shows the severeness of disease after infection by influenza, right? At a young age, if the person got imposed to H1N1, and there will be immune response developed to H1N1, right? The second time, if this person imposed to H1N1, the severity will be much lower because there's already some memory B cell, T cells to reduce the severity of the disease, right? And later, if there's a slightly different, this exposure to H5N2, right? There's one similar protein there. So there could be, might be cross protection because of similarity. But if there's a primary infection with H3N2, which is totally different virus, then this person will be primed to the first exposed virus for the rest of the life, but as a cost of maybe have a bad immunity response to another strain of influenza. So you would see a very severe disease, probably after infecting by another strain. So yeah, that's why, you know, a lot of people, they think about what the immunity landscape across different locations. They will look at the birth cohort when the people were young, what kind of virus they were exposed to, right? So this is figure from a review paper. This shows the time from late 19th century to 2018, and on top shows the major circulating strains of influenza, right? And you can see different people when they were born, they may experience different viruses, and they may have a better immune response later in their life to the first strain they were exposed. But if there's some other new viruses, virus strains infect them, they may have a bad response there. And that's why there's study looking at this effect. You see that maybe the middle-aged individuals may be in the perpetual state of H3N2 influenza virus susceptibility, because those people were born around this age, they were exposed to H3N2 at that time. But now I think the contemporary H3N2 is a little bit different from that. So they can respond pretty well to that strain. But if there's a new, for the contemporary H3N2, they may not, they have a lower, worse immunological response. So yeah, I think that's probably, I will stop here, and I think there's a lot of unknowns in the future about what will happen in terms of flu outbreaks. I'm just listing some of the literature and some of the papers talk about this topic, and I hope people here, they can, get interested in this topic and try to use data and modeling and different methods to study this and try to see whether we can help people to reduce the burden of influenza in the future. All right, I just recap what I talked about. First, we talk about the pathogen interactions, some of the methods used in the previous studies, and we look at the empirical evidence of COVID-19 pandemic during the last 3 years. We used a model study to showcase how we can quantify impact of NPIs on COVID-19 in the US across different locations. And lastly, we talk about several factors we still don't know the impact on future influenza outbreaks. So I think I'll just stop here and take some questions. Thank you so much.
Sen, thank you very, very much. I was really intrigued there when you were kind of touching on some of the kind of the genetic influences that may be affecting the viruses, making them more susceptible or making people more susceptible to them. And I noticed in several of the studies that you were talking about kind of correlations between viral outbreaks or viral exposures, where there was kind of a temporal, as one peaked, so did another one, or rather one peaked and another one went down because people weren't as exposed. I was kind of curious whether or not there is, over a long time, as these viruses kind of are in the population with those things happening, are there any trends towards like co-evolution of viruses that kind of make either of them stronger? Do they reach any sort of an equilibrium where they just kind of are constant in the population at particular levels? Or are there any opportunities for viruses and the culture of the people that they infect to co-evolve as well? Does that make sense?
Yeah, I think that makes sense, but I haven't seen a lot of studies on that topic. I mean, because usually there are a lot of factors shaping the evolution, and and also the transmission at the population level, right? I think the key point here is we are lacking of those high resolution data to answer those questions. If someone can provide maybe individual level data, longitudinal, so you can better understand what's going on there. I think definitely we can use a mathematical model to look at whether there will be a steady state or whether there will be some pattern there. But it's very important to use those detailed data to answer those questions, right? Yeah,
I agree. And one of the things I was kind of puzzling on while you were giving your presentation is you had a couple of maps that are showing the maps of the United States showing the outbreak concentrations for these various viruses. And I was kind of wondering, like when we were having the COVID-19 pandemic, the Johns Hopkins people got very famous for having their COVID-19 map and whatnot. And it occurred to me that, we have maps on our phones, right? And we get I've got a weather app, I've got Google Maps, I've got, several different mapping things. That why don't we have like a virus map that's really anywhere you are in the world, much like Google Earth or Google Maps, you'd be able to go and see what the weather report is, the viral weather report for your particular area. Anyway, while you were talking, I went and looked it up. I found this one called Flu Star, which is an app that you can get for your phone. But it's still kind of low res, but you can also imagine that the viral transmissions and its locations and concentrations are very much like the weather. And as we move around, we're moving around in the space that the virus moves around into, much like, we can expect tornadoes in Oklahoma and we could expect a hailstorm somewhere else, that these would be different forms of viral weather, if you will. And I'm curious if you think that there might be something like that for the entrepreneurial minded, maybe they could pursue that.
Yeah, I think that's the question relevant to disease surveillance, right? So currently the surveillance mostly come from healthcare systems. You go to hospital, you get tested, they will report to the CDC or not. I mean, so this makes the whole process very slow. I think the reporting for flu have a lag of 2 weeks. You don't get the real-time data. Yeah. So the other option is citizen science. I think there's some apps called Flu Near Me. So you can report symptoms or any people infected by some viruses, right? So you can report those to there. So you will see what the general prevalence across different locations, but that has a caveat because you don't have the laboratory test, right? You don't know what virus it is. You can only know this person has a cold. Could be COVID, could be flu, could be other things. So yeah, and also I mean, there's really popular to have some like mobile devices people can track. You have those fancy Apple iWatch, right? If you can use those devices to collect data, that would be fantastic. But I think the key point here is whether do you want to know the resolution of which virus causing the illness. If you only want to report illness, I think it's possible to do as long as a lot of people want to do it.
Yeah, it's funny. I was thinking as you were just talking about the one where you can report, people who may have the flu near you. I could see the downside of that is that it'd be like swatting your neighbor, right? If you didn't like them, you could that person has the flu, and you could report them and be mean to them. But on the other hand, I was thinking that another way that one could enact something like this would be to put monitors in, say, monitoring sewer systems, for example, where you could actually detect the presence of the virus or which virus it is, which was present. And, most places with a sewer system would be able to, if we had a device that you could install, and it would then report the presence or absence of various viruses and whatnot, that would at least give us a hint of what's out there and where is it and in what concentration and what to watch out for. It could be done in real time without having to ask people.
Yeah, and it's faster. I mean, wastewater surveillance is very important right now. I think starting from COVID, and people doing a lot of those. And actually, there was a paper published. I was thinking to put it here. They look at the wastewater data. They sequence. They look at a bunch of respiratory diseases in the data. And they found some of the common respiratory viruses was the RNA copies are lower during the COVID-19 pandemic. It's just showing that this could be a possible way you can detect. It's passive. You don't need to get consent. It's earlier than symptoms, right? You don't have to wait until there's a symptom. And there's a lot of advantages there, but also there are some challenges for that because you have to link. I mean, if you only need to know whether there is or not, it's easy to do, right? If you find it, there is it.
Yes.
But if you know the quantity, what does this number mean at the population level? It's a little bit challenging to do that because all the factors coming into that.
Yeah, and I'm thinking that the modeling becomes so much more than just looking at a time course, right? You're also looking at the change in space of where's the concentration peak at that particular moment. And it's going to move, right? In which case, it probably has to be something like you're solving a second order partial differential equation or series of them in order to model the patterns of the interactions between the viruses as well, in space as well as time. And that becomes very computationally challenging. And you're absolutely right. If there's people in the loop who have to give consent or have to be monitored, by the time they're reporting it, you've missed a lot of time. It would need to be something that one would have at, you'd have in wastewater locations, you'd have samplers that would report in real time. And much like a weather station on the top of a mountain someplace, scattered around the country, one could then interpolate between them and use them as fixed points for, a partial differential equation modeling.
Yeah.
Of course, it sounds easy when I say it, but I'm sure that it's a lot harder than that. What sort of skill sets do you see data scientists needing to be able to help develop these models and make them something that could be actionable like this?
Well, there are a couple of things. If you want to focus more on the the interaction part, I think data science should be able to understand some of the typical methods you statistical methods to disentangle interactions between time theories. Those are the techniques I think is important to know. But also I think it's important to have some biological background about how those pathogens interact, what are the potential pathways to do that. And if you want to do something operational, you want to push something to use in real world, then that's a big question because you have to understand all the limitations in implementing those things, like what the data limitation, there will be reporting delay, there will be missing data, there will be, revising, it's a lot of uncertainty coming into this. And also how you can try to convince people to use it, especially how do you work with maybe public health agencies. Those are something pretty important. But I think this exercise is kind of interdisciplinary. It really depends on which perspective you want to focus on. But I do think it's important to know both the data and the biology and of the question.
I think that's super important because the accuracy of your model, you may be able to get some numerical accuracy, but there's also the kind of construct reliability of the measurement, right? Is it actually reflective of underlying biology, or is it some, epiphenomenon and you're just really good at detecting it?
Yes, exactly. Yeah.
Any parting words about, what's coming next for this kind of modeling of flu or COVID or other viruses that we have yet to be exposed to?
I don't know, this is a pretty big topic, right? So a lot of people actively working on this, and involves a lot of fantastic research questions. I think a lot of people are talking about involving human behavior into disease transmission, right? So how do you, because a lot of those interactions I described was mediated by human behavior. You reduce contact, then you will reduce the transmission of multiple pathogens, not just the flu or COVID. I think that's something, if you can get a better measurement of how people interact, how people stay together, and without violating some privacy issues, that will be very, very helpful. And I do think there's some data starting to do that. For other questions, because I'm not working on like real biological questions and all those immunological questions, I probably should not talk about those topics. But I think mostly in my research, in terms of data science, I think measurement of human behavior is very important, and how you use that data to inform disease transmission and evolution. That's very important topic right now.
That's fantastic. Well, Sen, thank you so much for sharing your work with us and these ideas about viral transmission, not just COVID, but those other viruses, those pesky viruses which are out there that we're sure to experience once again here once this summer is over and flu season comes up again. But thank you so much for sharing this. Thank you so much for giving us some insight into how best to model this. And your work is fantastic. Everyone have a wonderful, wonderful weekend. And I encourage you to tune in next week when we have our final biomedical data science seminar series. And we'll see you then. Have a wonderful weekend. Thank you again, Sen.